So there are a lot of inflammatory mediators that exist. There are a lot of just immune chemicals that exist. It's its own branch of science, its own branch of study. And unless you're kind of an immunologist and you spend your whole life kind of studying these chemicals, it can be kind of difficult to keep straight what's doing what. So in big, broad, general terms, we're going to go over some of the most important of these inflammatory mediators, what produces them, and kind of what their role is. First off, we'll start with the inflammatory mediators that are produced by cells. And of our cells, we've got our white blood cells. So these are, again, our leukocytes. Searching back to 228, our different leukocytes are things like neutrophils. I'm just going to do first letters. Neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Just in brief, if you remember, neutrophils are kind of the first responders. They arrive to the area of tissue damage first, and they die first. Eosinophils are mainly responsible for parasitic infections, but they're also associated with allergic reactions. Basophils release histamine. They're part of this inflammatory response. Monocytes are our precursors to macrophages. Remember, macrophages are our big eater cells, macrophage. They're some of the best phagocytes that we've got. Neutrophils are also phagocytes, but again, they're kind of short-lived. Lymphocytes are our T cells and B cells. Now, all of these white blood cells have their own types of cytokines that they release, and those cytokines are going to do specific functions. The inflammatory cytokines, those that are going to be our inflammatory mediators, help this process, are mainly released from basophils and mast cells. Basophils, again, produce histamine, but they don't only produce histamine. Histamine is a good vasoactive chemical that causes dilation of the blood vessels, which then leads to the swelling. Histamine is probably one of the most well-known of these inflammatory mediators, but basophils are also going to release prostaglandins and our leukotrienes. Again, we're just going in broad generalities here. Prostaglandins and leukotrienes are lipid-like molecules. It allows for stimulation of internal receptors, so we get a different response in response to prostaglandins and leukotrienes. Platelets are next. If you remember, platelets are the little cell fragments. Their main role is in the formation of clots, in the induction of the clotting cascade. Now, platelets are chock full of little chemicals inside, lots of different cytokines. Some of those cytokines are things like serotonin. Oh, no E on serotonin. Serotonin is generally thought of as a neurotransmitter. It happens in neurons that are connecting to each other and talking to each other. But serotonin also functions in our blood vessels as a vasodilator. Additionally, platelets can also release histamine as well. Lastly, we're going to focus on the damaged tissues in the endothelium themselves. So again, this is the endothelial cells, part of the blood vessels, and then our damaged tissues here will also release certain inflammatory mediators. Now, there's a lot that damaged tissue can release that's pro-inflammatory. Things like potassium, things like ATP, stuff that's normally inside of cells that shouldn't be in the extracellular fluid. When those cells are damaged, they kind of rupture and release that internal content. Now, those do work as inflammatory mediators, but there are very specific cytokines that these cells also will produce. So endothelial cells, damaged tissue, can release a cytokine called platelet activating factor. PAF. Platelet activating factor does what it says. I love when things are named like what they say. It does activate platelets. It also induces vasodilation. So it's going to help to initiate the clotting cascade, but it's also going to get platelets involved, releasing their serotonin and histamine as well. Injured cells can release arachidonic acid. I'm just going to title that AA. Arachidonic acid is a precursor molecule that can be converted into a whole bunch of different inflammatory mediators. And those different inflammatory mediators will have a different effect on inflammation. So I don't want you to panic when you see this. We're going to go over briefly what arachidonic acid can do. You do not need to memorize this. This is mainly to show you the interconversions between arachidonic acid, and it's going to be important when we talk about how we can fight inflammation. So arachidonic acid is a lipid. It's an eicosanoid, this 20 carbon chain molecule, and it's normally part of our phospholipids. When a cell is damaged, a certain enzyme called a phospholipase will go into our phospholipids and cut off one of those fatty acid chains, and we get something that looks like arachidonic acid. Now, arachidonic acid will be converted into a different inflammatory cytokine depending on which enzymes it encounters. So if the COX enzymes are present, this stands for cyclooxygenase, this arachidonic acid is converted into prostaglandins. And prostaglandin is a big, broad, general term for a whole family of prostaglandin molecules, all of which have at least some play into inflammation. Prostaglandins do a lot in the body. It's a whole area of research. In fact, there's very little that prostaglandins don't affect, but in general, for what we're talking about, prostaglandins are big for the inflammatory response. Not only do they initiate it, but they also help to sustain it in a whole bunch of different ways. Then, depending on which types of enzymes are present, that prostaglandin can be converted into things like thromboxane. Thromboxane is a potent hormone for the clotting cascade, and it helps to activate platelets. But again, just another inflammatory cytokine. If our LOX enzymes are present, arachidonic acid gets converted into leukotrienes. Leukotrienes we've talked about a little bit. Again, they're just a general purpose inflammatory mediator that helps with vasodilation, bronchodilation, just sort of immune response in general. Lipoxins can also be converted from our leukotrienes. I don't know that we've mentioned those at all, and maybe we won't spend a ton of time on those. Again, don't memorize this. This is just showing that arachidonic acid is a very interconvertible molecule. It can be turned into prostaglandins or leukotrienes or thromboxane, all of which are going to have specific and general inflammatory responses. So those inflammatory mediators were all released from specific cells, and those cells usually are in the site of the damaged area, or they're immune cells that have made their way to the damaged area. But there's also inflammatory mediators that are constantly circulating in the blood plasma. Some of these are proteins that have been produced by the liver and are just kind of circulating inactively, waiting to be activated. Some of them are going to be released by immune cells that are distant. So let's start off with complement. Complement is a series of proteins that are produced by the liver in inactive form. There's about 20 to 30 proteins. And these proteins, once they become active, have a whole bunch of effects on the immune system. So part of the complement system is that it actually punches holes into bacteria by these assembling proteins, making their way to the cell walls and actually inserting themselves in the cell walls of bacteria. That causes pores to open up. Water will then flow into those cells and they can lyse as a result. Other parts of the complement cascade is that it increases the phagocytotic properties of bacteria. It makes them, complement acts as an opsonin, if you remember that term, making stuff tasty. It makes it easier to phagocytize bacteria that are covered in complement. But complement also induces inflammatory responses. So the complement system is important as one of these inflammatory mediators. The clotting cascade. So in the clotting system, remember, these inactive proteins then become active, leading to factor 10 becoming active. Factor 10 activates other proteins. Eventually, we get the conversion of fibrinogen into fibrin, and fibrin forms little subunit molecules that polymerize into large chains. And so we get these chains that act as the clot, which is part of the hemostasis regulation, preventing blood loss and fluid loss. Now, while it's doing the clotting cascade, it's also inducing inflammatory responses. And then the one we skipped, the kinin system. Kinin system, things like bradykinins. And I know this sounds kind of non-specific, but talking about the immune system, there's a lot of redundancy. A lot of these hormones do the exact same thing in slightly different ways. So kinins, bradykinins, are released from mast cells, and they kind of just float around in the blood plasma. The kinin system is implicated in things like asthma, respiratory responses to allergic reactions, septic shock. That kinin system is important and it does a lot, but for our purposes, it's part of this inflammatory response. All right, we're going to jump back up a couple of slides just to go over that flow chart one more time. So we've gone through the different cytokines that are released, what their kind of role is in either being a vasoactive mediator. Now we're going to talk about some of the chemotactic mediators, things that bring our white blood cells to the area. So chemotaxis is the process of cells moving from low concentrations of hormone substance to a high concentration of hormone substance. Specifically, we're talking about chemokines. Chemokines are a type of cytokine that's responsible for directing movement of cells. And how this works is if we've got an area of damage, it's going to start releasing these chemokines, and then our immune cells will encounter those, and they'll make their way towards the higher concentration. It might be a little hit and miss. They might come over here, and then, oh, that's not right. Concentration decrease. Oh, eventually, they make their way to the source. This allows for our leukocytes, immune cells that aren't present at the actual site of location, to then make their way to that site, and then have their effect specifically once they get there. Parts of the cellular response here, chemotaxis, so that's when these immune cells first encounter the chemokines and they start making their way towards the higher concentration. Once they do that, they're going to come in contact with the endothelial cells, and they'll start adhering to the sides of the blood vessels. So instead of floating directly in the blood plasma like our red blood cells would, once they come in contact with chemotaxic molecules, those chemokines, they will attach themselves to the wall, and then they'll start rolling so that they don't accidentally miss where the concentration of chemotaxins are coming from. This is called cell adhesion, so they'll bind to the endothelial cells with specific proteins, and then they'll just roll until they get to an area of high chemokine concentration. Once they get to that area, they're going to induce a couple of cellular changes, move proteins around inside, and that's going to allow them to squeeze through the gaps between our endothelial cells. So these capillaries have now become pretty leaky because a lot of our vasoactive chemicals have caused the connections, the desmosomes, tight junctions, to break apart between our endothelial cells. These white blood cells can then squeeze their way from the vasculature, from inside the vessel, out through a process called cellular migration, or diapedesis is another term for it. Cellular migration works just fine, though, as a term if you want to use that one.